All right, the third molecule or macromolecule we'll talk about are amino acids, okay? And so amino acids are, their composition is they will have an NH2 that is combined with an R group. An R group is a carbon-based group, which we'll talk about in just a minute. And this R function, the functional group, actually tells me which amino acid I have, of which there are 20 of them. And then there's a C double bonded OH at the very end. Again, the R group determines the amino acid, and the protein is determined by the different amino acids and how they come together. As always, we're going to draw a diagram in a minute that's going to go right here, and you're going to write really small and really small so you can fit the monomers over here and the polymers over here. But when we create a bond, we're going to do dehydration synthesis. The type of bonding here, different kind of name, it's called a peptide bond, okay? And if we were to go the opposite direction and go this way, we would do hydrolysis or hydrolysis, okay? And that's that. Okay, on the left-hand side now, what you're going to want to do is you're going to want to write in these two monomers. This is just the left-hand side. You can see the arrow right here, okay? And there'll be more amino acids. So this is just 1, 2, but you can have a whole bunch of amino acids put together, okay? And so the R group, again, determines the amino acid, of which there are 20 of them, and it's this thing right here and right here. So that's why we just symbolize that with an R. Okay, but this is amino acid number 1, monomer number 1. This is amino acid number 2. You can see that symbolized by the plus sign between them. I actually recommend that you take a moment and hit pause and write this in. So hit pause and write this in on the left side of the arrow. These are the monomers that go into the reaction. These are the reactants. Okay, this page is just showing you an example of an amino acid. You actually don't have to draw anything here, okay? I just wanted you to see some things. So, alanine is an example, or threonine, or spartic acid, okay? Tryptophan. This one at Thanksgiving time really shows up for us. And then there's lysine. So these are just 1, 2, 3, 4, 5 examples of just some amino acids, and I just wanted you to be able to see them. Again, it's the R group that makes them different from one another. All right, so now you do need to write this out, okay? So the only thing you have to write here is this is the monomers again, okay? So you've already drawn this side. Here's your arrow, right? So I just want you to put a box around the 2 hydrogens here and the oxygen so you could see the water molecule that's going to be pulled out. And now the right-hand side, the polymer, okay? So this is called a protein. Proteins are determined by their amino acid combination. And you can see right here is where we pulled out that water molecule. So really, if I was going to do this, I should write plus H2O right there. So please write that in, plus H2O. So I want you to pause this, and I want you to record and write in this entire protein. Again, this is the product side, the right side of your drawing. Pause it, draw it in. All right, and from here, okay, so back in your notes here. All right, so what type of bonding is this? Okay, the type of bonding that we have here is called a peptide bond, so that's that. If we're going to break the bond, anytime we break a bond, break things down, we do a hydrolysis. Now, last thing, okay, is what is a polypeptide? Well, anytime we see poly, we should think many or more than one. Peptide means the type of bond that's here. And so what is a polypeptide? It's a protein that is made up of many amino acids that are bonded together. And an example of a protein that comes across a lot for us is something called an enzyme. I've referred to some enzymes to you, things like maltase. So anytime you see ASE at the end, it means it's an enzyme. And they're proteins, okay? And so proteins work like this. So if you remember the other day, I told you about how your body can break down starches, but it can't break down cellulose, right? That corn comes out exactly like it went in. And that's because of this thing called amylase. And so the best example, or the best way to explain this, is something called the lock and key hypothesis. And so amylase, if you look at it, right now I have like a Pac-Man here. And so this would be like the lock, right? And so as you know, if you go to like enter your friend's car, you can't get in because you don't have their key, right? And likewise, they couldn't get into yours. And that's because certain keys are specific to certain locks. Well, here's how this works. Amylase has this chunk that's missing right here, right? And so what it's looking for is it's looking for something to complete itself. It's called an enzyme amino acid complex, okay? And so what happens is it's looking for the starch that matches together with it. And so this starch, as you can see, would match up right here, but it wouldn't match up with something like cellulose. So that's why amylase is specific for starch, not for cellulose. And that's true of enzymes. The biological application of proteins, they're found in your skin, they're found in your hair, they're found in fingernails and muscle, and in enzymes.